Hey everyone, and welcome. When you think about a biologist lab, you probably picture a microscope and petri dishes, right? Well, these days, the computer keyboard is just as crucial. Today, we're going to dive into how programming has become a flat-out essential skill for modern biologists, totally changing how we decode the very language of life. Alright, so here's our game plan. We'll kick things off with the why, this massive data problem that really started this whole revolution. Then, we'll check out the go-to tools for coders in biology, Python and Julia. After that, we're going to take a peek into the future. And it's wild. Think AI meeting DNA. And finally, we'll lay out how you can start your own journey into this world. So, first things first, biology's big data problem. You see, our ability to sequence genomes and analyze entire biological systems has just completely exploded. We're not talking about a little more information, we're talking about a tsunami of data. Data sets so huge that trying to go through them by hand is, well, it's just impossible. And this quote right here just nails it. When a single experiment can spit out terabytes of data, speed isn't a luxury anymore. It's the absolute difference between a breakthrough and a dead end. Computational tools aren't just kind of helpful now, they are completely and utterly essential. For the longest time, scientific coding has been stuck in this classic dilemma, what people call the two-language problem. You either pick a language that's easy to write in so you can test ideas fast, but it runs slow, or you pick a language that's blazing fast, but it's a total pain to write. Scientists need both, right? They need to prototype and they need to run huge simulations. This constant, frustrating trade-off is what really drove the hunt for better tools. So what's that first tool that most biologists grab from their digital toolkits? 9 times out of 10, it's going to be Python. It's really become the Swiss Army knife for this field because it's just so incredibly versatile. So why is Python so ridiculously popular? Honestly, it's because the code itself is designed to be super readable. It almost looks like plain English. This is huge because it lowers the barrier to entry and lets scientists focus on their biological questions, the stuff that actually matters, instead of wrestling with super complicated code. And you can see that beautiful simplicity right here. A super common task, like reversing a DNA sequence, maybe to look at its complementary strand, takes just one single clean line of Python code. That's it. But don't you dare mistake that simplicity for a lack of power. Oh no. When you add in specialized toolkits called libraries, like one named Pandas, Python turns into an absolute beast for analyzing data. For instance, with a command just like the one you see here, a biologist can take a massive file of gene data, instantly group it by cell type, and find the most active gene in each one. That's a super complex task made incredibly simple. So here's the crucial thing to understand. The basic Python language is great. It's the engine. But its real power, especially for biologists, comes from its huge ecosystem of libraries. It's like having a garage packed with specialized tools. You've got things like BioPython, made just for handling DNA sequences, and then heavy hitters like Pandas and scikit-learn for world-class data analysis and machine learning. Okay, but for all its strengths, Python can still chug a bit when you throw really intense calculations at it. And this brings us to a newer player on the scene, a high-performance language that was built from the ground up to solve that exact problem, Julia. I love this book because it perfectly sums up Julia's whole deal. It's got a clean, easy-to-read syntax that feels a lot like Python, so it's comfortable to write. But under the hood, it's an absolute speed demon with performance that gets close to old-school languages like C++. It's a direct shot at solving that two-language problem. Julia gets its power from a few key features. First, of course, is raw speed. Second, something called abstraction, which is a fancy way of saying it's great at letting you represent complex things, like biological systems, in a really intuitive way. And third is a real mind-bender called metaprogramming. This is basically code that can write other code. It lets you create powerful, custom tools on the fly to solve your very specific research problems. And this right here is a perfect example of Julia's cleverness. With a feature called lazy operations, you can line up a whole chain of complex commands. But Julia is smart. It just waits. It only does the actual number crunching at the very last second, when you actually ask for the final result. It's a brilliant way to avoid tons of unnecessary work. Alright, now let's jump to the absolute cutting edge where this is all going. We are moving into a brand new era where we can use artificial intelligence, specifically large language models, to learn how to interpret the language of DNA itself. The shift in thinking here is just profound. Instead of just counting up A's, C's, G's, and T's, scientists are starting to treat DNA like a language, with its own grammar and meaning. They're taking AI models that were trained on the entire internet and retraining them to read genetic code to figure out what it does, like identifying which sequences act like on-off switches for genes. So how does that even work? Well, a simplified version looks something like this. First, you need to translate the DNA alphabet into numbers that the AI can actually crunch. That step is called tokenizing. Then you set up the model itself. And finally, the really cool part, you fine-tune it. You train that big general AI on a very specific biological task, teaching it a new skill, like how to spot a landing pad for a protein on a strand of DNA. There is one huge problem, though. These AI models are gigantic. They need a mind-boggling amount of computer memory. And that's where a really clever trick called quantization comes into play. It's basically a way to shrink the model down by reducing how precise its numbers are. This makes it possible for a researcher to run these incredibly powerful AIs on a computer in their own lab, instead of needing access to a supercomputer. And that really gets to the heart of why this matters so much. By taking existing models and just fine-tuning them, we slash the time and the cost needed to do this kind of research. It's a huge step in democratizing AI. It's putting these state-of-the-art tools into the hands of way more scientists everywhere. Okay, this is all incredibly exciting, right? You're probably sitting there thinking, how on earth do I get started with this? Well, the great news is there have never been more high-quality, low-cost ways to learn than there are right now.
For example, platforms like Kaggle Learn are amazing because they get you working on real-world projects with real data right from the get-go. If you prefer a more structured, university-style approach, Coursera is a fantastic choice. And if you're someone who learns best in short bursts, DataCamp is perfect, with its bite-sized, interactive lessons that give you feedback as you code. So, all of this leaves us with one final massive thought. We now have tools that can analyze data on a scale we could barely imagine before. We're even teaching AI to read the code of life itself. The Digital Biologist's Toolkit is more powerful than ever. So the only real question left is, what biological mystery are we going to solve next?